Classic theory predicts that new species may form when once closely related populations stop interbreeding long enough to independently accumulate unique genetic changes that would otherwise be incompatible in their hybrid offspring. And evolutionary biologists have long wondered which genetic changes result in such incompatibilities, and if those follow predictable patterns. Hi there, I'm Natalie Gonzalez, a PhD student in the Department of Genetics, and I'm using several species of yellow monkey flower to try to answer some of these questions. Hybrid seed lethality is common among crosses of flowering plant species, and it often results in flat, inviable seeds. It describes incompatibilities resulting in developmental failures of a very critical seed tissue called the endosperm. The endosperm tissue facilitates nutrient transfer from maternal to embryonic tissues, and in this way it very much acts the same way as the mammalian placenta. Endosperm development also requires the fine-tuned regulation of many genes, some of which are turned on or off depending on whether they were inherited maternally or paternally. This parentally biased gene expression, known as imprinting, is important for development, but it may also serve as a source of genetic incompatibilities between divergent plant species. Few imprinted genes have been described outside of model organisms, and it's unclear how conserved these genes are among flowering plant species, including some used in agricultural research. My research aims to identify the incompatible genes causing hybrid seed failure in monkey flowers and to determine if those genes are imprinted. By using multiple species pairs, I can determine how conserved imprinted genes are among several closely related plant species, and, hopefully, I can also provide evolutionary biologists a better understanding of the role of these genes in the formation of new plant species. By crossing a few surviving hybrid seeds to their parental species, I've already generated hundreds of plants. These surviving plants, in theory, should lack incompatible genes, and I can sequence their DNA to identify which genes are missing among my hybrid populations that were present in the original parental species. In this way, I can develop a list of candidate genes from the parental species that are incompatible. Then, I can determine if those genes are imprinted by assessing gene expression in healthy endosperm. For this, I will use RNA, which comes from active DNA as expression products. By dissecting out endosperm from viable seeds and sequencing their RNA, I can identify which genes are imprinted and determine if they're among my gene list from the DNA experiments. My research findings may broaden our understanding of how imprinting contributes to some of the genetic incompatibilities that eventually lead to the formation of new plant species. I want to conclude this video by thanking the ARCS Foundation reviewers for considering me as a semi-finalist for this scholarship.